This one, the genome sequence of Thermotoga maritima and evidence for lateral gene transfer. This was an early genome sequence, and I used this just to kind of show you how where genome sequences have come. It's from Karen Nelson at the Institute for Genome Research. At the time, genome sequencing was pretty early, and so a lot of this paper is about the nuts and bolts of sequencing and sorting out the data. You can look at the GenBank file for this, and you can do all the visual searches for the genomes that you want to do. Nobody publishes this kind of information anymore, but I think it's interesting just to show you what's in a genome. The length of the sequence is 1,860, 725 nucleotides. Yeah. Because you have it as a separate thing, an online supplemental information or something like that. It's trivial. You don't learn anything from that. 46% GC in the genome overall. The sequence was assembled from 30,140 individual sequence reads, which averaged 531 nucleotides in length. They found 1,877 open reading frames, i.e. probable protein coding genes. And those open reading frames cover 95% of the genome. And so all the other stuff, promoters, terminators, RNA encoding genes, origin of replication, terminus of replication, all the other stuff is encoded in the remaining 5%. There is one ribosomal operon, and it is not in the usual conformation. So ribosomal operons in bacteria are almost always 16S followed by 23S followed by 5S in that order. Usually there are a couple of tRNAs embedded in the spacers. In this case, the 5S RNA gene precedes the 16S gene instead. There are 46 tRNAs. 19 of them are in individual genes, and then there are 10 clusters of them. Clustering of tRNAs is common. Of the 1,877 open reading frames, 1,000 of them showed similarity to known proteins at the time. This would be increased by now, of course. This is how you identify the function of genes, right? You look to see what it's similar to in sequence, and if it's 95% similar to an alcohol dehydrogenase, it's probably an alcohol dehydrogenase. And so 1,000 of them are similar to known proteins. I'll point out that they don't always know exactly what the function is. It's quite common, for example, to know that you have a dehydrogenase, but you don't know what the substrate is. And so you don't know if it's an alcohol dehydrogenase or something else. There are 407 conserved hypotheticals. What's a conserved hypothetical? This means that it's a gene that's found in other organisms, but the function of it is unknown. And these are quite common. There are a bunch of genes that we just, even today, just don't know what their functions are. A lot of times people will, so for example, they walk through the entire yeast chromosome and delete the genes one at a time to see what their function is. The problem is twofold. One, you get lethals, right? If they're lethal, you don't learn anything. You just know it's essential, but you don't know what it does. Sometimes you can get an idea by not deleting it, but regulating it. And so if you turn it down, you get a phenotype. It doesn't quite kill them, but you start to see defects, and you can sometimes get a clue. In other cases, they delete the gene, and they get no phenotype. The cells look perfectly normal. But if they're conserved genes, they've got to be doing something. Just not something essential under the conditions that you're growing them under. An example of this might be ampicillin resistance, right? I mean, ampicillin resistance is in lots of organisms. If you delete it, nothing happens, unless you grow in an ampicillin. There are 83 of them that are similar to proteins of unknown function, and 373 of which have no database mapping. These should add up more or less to that. There are repeats in the sequence, and we'll get to why this is important. One of them is a 30-mer sequence shown here that's present in 143 times in the genome. These kind of repeated sequences are common in organisms that pull out DNA out of the environment and recombine them into their chromosomes. Haemophilus influenza is another organism like this. These are organisms that are designed to pull new functions out of DNA in the environment. And then there's a bunch of them that occur twice in the genome. And some of these are pretty obvious. You've got an alpha-glucosidase. It's just a gene duplication, right? You've got two copies of it. Two copies of a methyl-accepting chemotaxis protein. But most of them are transposon-associated proteins. And so these are transposons that occur more than once in the genome, or chunks of transposons that are scattered around the genome. Transposase, another transposase, a helicase. Helicases are used as part of the transposition process, right? You separate the strands as doing part of the replication. Likewise, an exonuclease. And so these are probably primarily transposase-associated enzymes, transposon-associated enzymes. Here's a map of the genome. How many of you have seen this kind of thing before? Most of you, I assume. Yeah, great. So the outer two circles are the two strands, the plus strand or the minus strand, showing the open reading frames that are encoded on each of those two strands, and they're color-coded based on this classification of their function, things like nucleotide biosynthesis, amino acid biosynthesis, translation, transcription, etc. And you can see there's a bunch of genes all over the place. I'll point out that the number one, is arbitrary in this genome. There's no definition of one base versus the other. Yeah?
We see a lot of white spaces of this what we don't know.
So some of it is, so for example, this white space here, because all the genes are on the other strand. And so, in general, wherever you find a band on one strand, you won't find on the other, because in general, there isn't a lot of overlap between protein-coding genes in bacterial genomes. There is a couple of nucleotides. Once in a while, there's some interesting overlaps, but not usually. That's in contrast to a lot of viruses, right, that have lots of overlapping genes. Yeah, smaller genomes. That puts a huge constraint, an evolutionary constraint on those sequences, because you've got evolutionary pressure, usually in two different directions on the same nucleotides, and that creates some complications. What else? This inner circle here, the next one in, let's see, second circle from minus strand, chi-square composition of, okay, so this is base composition. So, in other words, if you've got an overall genome of I think it was 40, what is it? What's the overall percent GC? 46% GC. What these bands are places in the genome where they're statistically significantly different than 46% GC. That is, they're highly enriched in G and C base pairs or highly depleted for them. This is an argument they're going to make later that these might have been acquired from someplace else. These might be horizontally transferred genes. I'll point out that those clusters are highly concentrated right here. That's because the origin of replication is right there. There's a whole GC bias thing in the genome. Let's see, fourth circle, archaeal-like sequences. So these are sequences that are more similar to archaeal genes than bacterial genes. Again, an argument that they'll use that these genes are acquired horizontally, and I'll show you that that's not necessarily true. Small repeats. This is the sixth circle, large repeats. That's these ones. Let's see, 1, 2, 3, 4. 1, 2, 3, 4. Fifth circle, large repeats. Seventh and eighth are ribosomal RNAs and tRNAs. So here's the ribosomal operon, and here are all the tRNA genes. If you look at this paper, there's this giant graph. I'm not going to show you in specific, but this is an eye-opening view of a microbial genome. That's it. And every one of these arrows is an open reading frame. The resolution on the screen probably isn't going to do this justice, but no. Every one of these arrows is a protein coding gene. If you've never sorted through a genome, you have no idea of how complicated these things are. These are pretty simple organisms as organisms go. I mean, a genome of less than 2 megabases. There's a lot in there. And again, this is the same color coding scheme that the circles show. Here's a table of all the genes. And so here we get a view of what this organism can do. And so, for example, there are genes for aromatics, tryptophan, alanine, biosynthesis, aspartic acid, and so forth down the line. You don't need all the genes to show what an organism can do. Remember, just over half of these genes are identifiable. But if you've got a handful of genes in the tryptophan biosynthetic pathway, you can be pretty confident that it can make tryptophan, even if you can't identify all of them. Not absolutely sure, but pretty sure. It's got genes for making and recycling purines, pyrimidines, nucleotides, and nucleosides. It's got genes for fatty acid biosynthesis and for phospholipids and for turning them over. It's got genes for some cofactors, biotin, folic acid, heme, porphyrin, quinones, pantothenic acid, riboflavin, and so forth. Not all organisms can do this, right? Can you make pantothenic acid? Nope. That's why you have to have it in your diet, right? It can make amino sugars, and there's a bunch of those. Let's see. It's got genes for energy metabolism generally, hydrogenases and that sort of thing. It's got dehydrogenases. The electron transport genes are interesting. So it's got a cytochrome C. It's got some flavin and iron sulfur protein stuff, ferredoxin, but it doesn't have any of the standard electron transport chain proteins. So there's no evidence that this organism can carry out the usual complex electron transport system. It can only do rudimentary electron transport. It's got NADH dehydrogenases. It can do the interdeuterol pathway or glycolysis. Glycolysis is in here somewhere. It can carry out parts of the TCA cycle. Probably there are other genes in the TCA cycle. Biochemically, you can show that this thing has a complete TCA cycle, but these are the only ones where the genes can be identified. A bunch of polysaccharide genes. It's got a ton of transporters. This thing is a heterotroph. Thermotoga lives in solfataras and hydrothermal environments, but it's not an autotroph. It's a heterotroph. And so there's a bunch of ABC transporters for amino acids and oligopeptides. Notice that these aren't identified as specific substrates. You can tell they're ABC transporters. You can tell they're for peptides and amino acids, but you don't know what the specific substrates are. You would have to have a three-dimensional model and figure out what kind of peptide could fit into that to do it. You could, potentially. It turns out Thermotoga is a nightmare to do genetics in. Let's see. There's some ion pumps. Chromate transport. This is to pump out chromate. Chromate's pretty toxic, right? In a hydrothermal environment, there might be a lot of chromate, and so you have to be able to pump it out. Let's see. Maltose. ribose, and then a whole slew of sugar transporters, heavy metal transporters, ferritin for iron import, calcium channels, magnesium, ammonium. I'm looking for a particular one. There's an antibiotic efflux pump in here somewhere. Here we go, antibiotic ABC transporter. This thing's antibiotic resistant. That's pretty cool. In fact, there are several of them. A bunch of more ABC transporters of unknown substrates. It can do DNA metabolism. You know, it's got polymerases. It's got pol-3. I think that's the only pol this one has. It's got RNA polymerase. It's got, where are the transcription factors? Here we go. Sigma factor, 1, 2, 3, 4 of them. That implies that it can control gene regulation, right? Because of 4 different things. Let's see. It's got some process, and so the list goes on and on. Notice there's a bunch of tRNA synthetases. This is the stuff you need to live. This is what an organism has to be able to do to survive. It's got all the ribosomal proteins. It's got my favorite protein right here, RMPA, the dispensable protein subunit of RNase-P. It's got a bunch of other stuff. The list goes on and on and on. So let's take a look at what this means. Remembering that they only can identify a little over half of the proteins here, and even in those cases, a lot of times they don't know what the substrates are. They can put together a pretty good idea of how this organism makes a living. And a lot of this confirms things that people already know from growing it. It's a heterotrope. It pulls in peptides and sugars. It's got all the sugar metabolism you could imagine for processing those sugars. It can make most of the amino acids it needs. It can make purines and pyrimidines. It's got all the genes for making flagellar motors and flagella, and for controlling them. It's got the key system. Let's see. Here's some more sugar things. It's got an antibiotic efflux pump. It's got an ATPase. Notice that the ATPase is shown hydrolyzing ATP and pumping protons. This is because it doesn't have electron transport channels. Almost all of these transporters are ATP-dependent transporters. And so you're using ATP to pump them. But there are key ones that are proton-dependent. They run on the proton gradient. And those require a proton gradient that you make with this ATPase. There is another proton pump that's based on the hydrolysis of pyrophosphate. Where would pyrophosphate come from? Excuse me? Generation of nucleotides, but there's an easier answer right here. A modification of this can make AMP plus pyrophosphate, and pyrophosphate can go in here and you can pump 2 protons as a result. Here's the ammonium transporter, potassium, iron going in, sodium coming out, cations going out, iron going in, potassium going in. Organisms like potassium a lot more than sodium. Magnesium going in, etc. Talked about that. 4 sigma factors. Interestingly, there are 2 amino-acyl tRNA synthetases absent from this genome. Those for glutamine and asparagine. In other words, they can't charge those amino acids onto tRNA. That would seem to be fatal, wouldn't it? It turns out it's not. In a number of organisms, including this one, the glutamine tRNA is charged with glutamic acid. And then And amidase puts the amine on to turn that glutamic acid into glutamine. Likewise for asparagine and aspartic acid. Aspartic acid is charged on the tRNA, and then it's aminated to make asparagine. One of two things can happen then. That tRNA is ready for translation, right? And it can be used that way. If you're using those amino acids for other purposes, you can hydrolyze it off. And at that point, then, the tRNAs become part of the amino acid biosynthetic pathway. So you're just making the amino acid on the tRNA instead of making the amino acid and then putting it on the tRNA. This turns out to be common in some organisms, but hadn't been seen in bacteria before. The big thing about this paper, and even today, when you sequence a genome, you're not going to publish a paper that says, here's the genome of this cool organism. You have to find something to make the point of the paper. Very often, you don't know what that's going to be until you do the sequence, right? But once you've done the sequence, you've got to make a story out of it. And in this case, that story was evidence for horizontal transfer. And I urge you to go back to one of our lectures long ago where we talked a little bit about horizontal transfer. In this paper, they argue that 24% of the genes looked like they were horizontally transferred recently enough that they still look foreign. You might imagine that if a gene's been in a genome for a while, over time, it would begin to look more and more like it belongs. They argue that 24% of the genes were acquired recently enough that they can still point them out as foreign genes. It depends on the genes. Certainly, in the case of E. coli and salmonella, you can identify genes that have been in place for less than 100,000 years. Most people would argue that 5% is a more realistic number. But that's still a lot, isn't it? You can't ignore 5% of horizontal transfer recently enough that you can still identify it. And so we're going to argue here about how much horizontal transfer is going on. But we're not going to argue that they don't have any evidence at all. They do have absolutely clear evidence of horizontal transfer, just not as much as they claim to. And ultimately, their point is that this organism is an evolutionary chimera. It's a collection of genes. And from their perspective, and they talk about this in the paper, bacteria don't have a meaningful phylogeny. They argue that genes have phylogenies, but that an organism is just a temporary collection from the gene pool, and that changes all the time, and so there really isn't any such thing as an organismal phylogeny. I think that's a false view, but we'll talk about it all the same. So what evidence do they have of horizontal transfer? Number one, 24% of the genes are more similar to archaeal genes than bacterial genes. What does that mean? That means they took that protein-coding gene and they blasted GenBack, and they looked at the top hit to see if that was a bacterial gene or an archaeal gene. How can you go wrong with that? Yeah?
So to compare the homology genes, someone would have to have found the function first.
In this case, they don't have to. All they have to do, they've got an open reading frame, they might or might not know what it is. They're just looking to see whether it's more similar to a known gene in bacteria or in archaea.
Those known genes would have to have been identified.
Not necessarily, because by this time there were other genome sequences available, and they could be unidentified genes in that organism as well. So how can you go wrong with it? Here's a table of this. And so amino acid biosynthesis genes, they have a collection of them. 49 of them, the best matches were to other bacterial genes, but 20 of them were more similar to archaeal genes. And 2 of them to eukaryotes, and 1 of them wasn't similar to anything. How can you go wrong with that? Yeah?
Well, theoretically, there's nothing to say that this gene is an archaeal gene. It just says that this gene has this purpose, right? So you can't really say, even though it is more similar to the archaea could have gotten it from bacteria.
That's true. So you could argue that the horizontal transfer may have gone the other way.
Right.
There's a simpler explanation than that. And it has to do with where Thermotoga is in the tree. So here's this three-domain tree, right? Bacteria, archaea, eukaryote. And all of these are collapsed just because there's a bunch of branches there. But Thermotoga is a deep branch in the bacterial tree, and it's a primitive branch. It's short. And so the sequences you're comparing this to are either here or here. The problem is, if you assume a constant evolutionary rate, you can make the argument that it ought to be closer to something here. But these individual sequences are going to have various evolutionary rates in different branches, and they're going to be scattered around some distribution curve. And so some of them are going to be faster evolving in bacteria, and some of them in archaeas, and so forth. And so you can imagine that a gene that is slow evolving in bacteria might actually be more similar in Thermotoga and in other bacteria. But what about genes that are faster evolving in bacteria? It might actually be closer to an archaeal gene, just measuring branch length, right, than in other bacteria. And so these differential evolutionary rates can make a huge difference on where they come out. You would need to generate a tree and show where they branch with, not just what they're most similar to. Yeah, Ben.
So it's more like evolutionary parallel transfer.
In this particular case, we're looking at an organism, at a sequence that remained primitive in Thermotoga, but had a faster evolutionary rate than other bacteria. So it's a rate issue. And this could even happen in eukaryotes, right, where a particular eukaryote may be quite primitive, like Giardia, and you may get a hit on that if it's particularly slow in that. These are protein coding sequences, and so as they specialize in different functions, the rates are going to be different. Imagine a lysozyme gene. Well, a lysozyme gene is going to be pretty slowly evolving in most bacteria, but if it's involved in the life cycle somehow, it might evolve very quickly and push it out into a different part of the tree. So this argument doesn't hold any water. What's another argument? There are about 50 regions where the base composition is statistically significantly different than the rest of the genome. They go to great lengths to point out that one of these regions is the ribosomal operon. Well, that's no big surprise, is it? Because this is a thermophile. And so it's likely to have lots of GC pairs in its RNA encoding genes. How many sequences is that? Well, there's one ribosomal operon, and there are 46 tRNA clusters. And all of those are going to have skewed GC bias because they're RNA encoding genes. They're not protein encoding genes. There are going to be other sequences with unusual GC bias just because of statistical variation across the genome. Since this paper was published, one of the things that's been noticed is that GC bias is different in different parts of the genome. So if you look at the genome and you just plot GC composition in big windows as you scan across the genome, the center of the peak of GC richness is usually right at the origin of replication. And as you get away from that, the genomes get more and more AT rich until you get to the terminals. And so there's lots and lots of reasons why GC bias is different in different organisms, in different parts of the genome. There are going to be some, though. For example, a transposon is likely to have a GC bias that's different than the rest of the genome because that GC bias is based on the optimum for the organism the transposon came from. And when it jumps into a new organism, it looks different, and it will slowly homogenize. And so some of these are legit, and some of them are not. The other, they don't talk about this. They imply it, but they don't talk about it. But one of the best ways to look at foreign genes is to look at codon usage. How many of you know what codon usage is all about? Okay, good. So most amino acids are encoded by more than one codon. I think there's 6 for lysine, for example. In some cases, a single tRNA can decode more than one, right? Because of the wobble in the third position of the codon anti-codon interaction. But in many cases, that's not the case, that you have two different tRNAs that decode different codons for the same amino acid. Many organisms have pretty strong preference for one set of codons versus the other. So the classic example is lysine, AAA versus AAG. E. coli almost never uses AAA to encode lysine. It strongly prefers AAG. Why? Because it doesn't make very much tRNA to decode AAA. It makes quite a lot to decode AAG. And so when that AAA codon pulls into the ribosome stalls, waiting for a tRNA that can match to it and let translation continue. An easy way to bring ribosomes to a stop is to put two AAA codons right after the other. And E. coli ribosomes will grind to a complete stop and will not proceed from that. This is one of the major issues for making foreign proteins in E. coli or in any other organism, is that if it uses a bunch of bad codons, then it isn't going to make very much or any of that protein. And it's common in molecular biology to include a plasmid in your E. coli that encodes isoleucine, leucine, and isoleucine, leucine, and lysine, TRIL, because those codons are so poorly expressed in E. coli that you won't make foreign protein. And so you provide them a source of extra amounts of that tRNA so that the ribosomes can move. So if you look at a gene and it uses bad codons, you might imagine that it comes from some other place, right? That doesn't work. And it doesn't work because it depends on the genes. If a gene has to be expressed at very high levels, think ribosomal proteins, it's going to follow the codon preference for that organism very closely. Because ribosomes have to move quickly across those mRNAs. When it sits at a codon, it wants a tRNA right there. But proteins that aren't expressed at very high levels, think RNase P protein, or, I don't know, protein appearing biosynthetic pathway protein. It can afford to have less abundant codons in it. If you're only going to make 20 copies of that protein in the cell to get by at any particular time, you probably don't need very quick translation of the mRNA. Also, proteins that have a very long half-life, that stick around for a long time, you don't need to replace them very quickly. In fact, poor codons are used to control gene expression. I mean, you guys know about attenuators and that kind of stuff. That's how they work. And so you control expression of proteins based on how quickly the ribosome can move over them. And so it's certainly true that foreign genes will have inappropriate codon bias, but you have to scrutinize that gene and ask yourself, is this a gene that normally follows the organism's codon bias or not? All the same, when you're done with all those caveats, there are about 5% of the genes in Thermotoga that clearly come from somewhere else, mostly from archaea. And add to that the fact that this organism, based on these repeat sequences, and those foreign genes, those 5% of foreign genes, are mostly associated with these repeats. Those repeats are designed. The way they work is that the organisms pull DNA in from the environment. And if they have this repeat, it knows it's a relative, and so we'll just recombine that right into the chromosome. That's a way of carrying out intraspecific recombination. It's a way of sexual exchange of DNA, right? But that system isn't perfect. And very often you'll find a sequence in a genome that's close enough to this that it incorporates it even though it's not a perfect match and it isn't from the same organism. And if that gene provides a positive benefit, it will be retained and built upon. So despite the fact that this organism doesn't have 24% of its genes from somewhere else, it does have quite a lot. And they argue in the paper, therefore, that you really can't base any phylogenetic analysis on a single gene. And they're talking about ribosomal RNA, right? That you really need to look at the whole genome and see where different parts of the genome come from. It turns out, though, that that's maybe a little simplistic. It turns out that you can really divide the genome into two parts. There's the phenotype part and the central processing part. And so there are a bunch of genes that are associated with the basic central dogma function of the cells. DNA repair, replication, transcription, translation. None of these have an effect on the phenotype of the cell, right? These are things that any cell just has to do. There's not really any pressure for an organism to pull out a new DNA polymerase from some other one, because it's got one that works just fine. And it isn't going to change the phenotype of the organism. Those genes almost never are horizontally transferred. And those genes, if you create a phylogeny based on those genes, you get something that usually that's pretty consistent with the ribosomal sequences. Remember that these are protein genes, and all the caveats we talked about at the early for why they're not as good as RNA-encoded genes are still true. But in general, those central processing genes will recapitulate the ribosomal tree pretty accurately. In contrast, there are a bunch of genes that aren't in the central processing of the cell, but are directly involved in phenotype. Amino acid biosynthesis. What can you make and what can't you make? Electron transport. All the genes that are involved directly in metabolism in the cell. These genes are much freer to move horizontally. And the reason is pretty obvious, right? If you pull in a gene, for example, for nitrate reductase, all of a sudden you can grow by anaerobic respiration when you couldn't before. That's something that selective pressure can do something with. And so those genes are going to move around a lot. Antibiotic resistance, ATPases, all these things that provide new functions to the cell that direct the metabolic capabilities of an organism, they do tend to move around. And those are the kind of genes, if you look at the 5% that are horizontally transferred to Thermotoga, they're all in that category. And it seems to be a general rule. If you look at horizontal transfer in microbial genomes, it's those metabolism genes that are horizontally transferred, not these central dogma ones, with one exception. And these are the tRNA synthetases that I talked about. Because it turns out that there are a lot of antimicrobials that target those tRNA synthetases, and they're single polypeptides that don't interact with anything but tRNA, which everything has, and amino acids, which everything has. Okay.